Thanks, Lydia. That's all right. Yeah, and thank you for the invitation and setting up this wonderful meeting, Lydia. And also thank you for all the training that you have done in your other programs that so many have benefited from. And thank you, Angela. I did want to put up a picture of Ken Kennedy because I knew Ken back, around the turn of the century, and was quite inspired by our interactions in the beginning. Well, actually towards the end of the Human Genome Project. The last time I saw Ken, he walked into a restaurant and I bumped into him and I said, "How are you?" And he said, "Oh, fine. I do have pancreatic cancer, but it's one of the good ones." And then he was gone about a year later. For those of you who didn't know him, you see, this is a publicly available portrait he had, and it's kind of funny. You see this guy bending over the thing. Gives you an idea of his spirit, and I'm sure those of you who knew him miss him terribly. But what a great way to honour him with your institute. Genetics is in a bit of trouble, in my view. It's gone through periods of, when I say genetics in medicine, it's gone through, I guess like AI, periods of hype. Is this working? Yeah. Hype and sort of dearths, where we thought that medicine would be transformed by genetics. It's going through a bit of trouble now because the people who are actually affected most by it, the frontline carers, are starting to push back. As we tell them it's easier and easier to do genetic testing, they're actually getting more reactive and really questioning the real-time value of genetics and genetic screening in particular in the practice of medicine. And they've got very good questions to ask. And the bad answers to their questions are all because we just don't understand what every gene does. As at the beginning of the Genome Project, we said we are like a Swiss watch, and if we understand the role of all genes, we'll understand all the parts, we'll really build this model of how some aspects of life work. And we're just not there yet. So we love AI, we love the idea of AI, and like all of you, and as Lydia's wonderful comments pointed out, there's been a lot of hype about AI. See, this is the engram. Machine learning is a very popular word now. This is a couple of years old. I think most biologists have been subjected to the idea that, I call it the cauldron of hope. If you take all of these things about genetics and other aspects of health and simply put them in some sort of unsupervised learning package, you'll come out with a mature disease model. And that's just not what happens. So I think we've got the humility aspect of this and how hard it's going to be. Not to say we don't have some good examples of applied use of AI techniques for advancing biology. Our poster child for that is actually methods first developed by Kyle Farr at Illumina, who took a series of primate genomes and used those to establish what the tolerant genetic pattern in primates would be, non-human primates, and compared those to human genetic variation to make predictors of what would be tolerable variation or not in the humans. That's expanded greatly now by us and other individuals who've contributed to hundreds and hundreds of non-primate genomes to establish maps like this, where you can see these are pathogenic variants, and in this gene map, that high scoring region is sensitive to the maleffects of variation. These low scoring regions are not. So that's an example of using deep neural networks and a lot of genomic data and outcomes data to make progress, but it's actually, I think, an exception. So what I want to talk about is around this question of the big challenge of completing this disease gene catalogue so we do get to the place where we understand what every gene does, and recognise that what we're doing now is mostly qualitative. I think, genetics is interesting sociologically because it does have this element of purely qualitative science and an element of statistical and quantitative science. But the gene discovery has largely been in the former, and we need to push it to the latter using new quantitative methods. Now, those of you who are involved in genetics might say, that sounds silly. Haven't we been doing a lot of quantitative science in genetics, for example, with GWAS and large studies? And the answer to that is no, that's not really advancing discovery in the way that we want to advance discovery. It is using quantitative methods to understand aspects of genetics, but it's not doing what I'm talking about today, which is simply understanding what each gene does. Now, here's a genetics lesson for you who may not think about this stuff every day. Largely, and in part historically, we've divided genetics into the simple problems on the left there and these harder problems on the right. The simple problems on the left here are simple single gene diseases, like muscular dystrophy or cystic fibrosis, where if you have the gene mutation, you have the disease. If you have the disease, you invariably have a mutation in a particular gene. And those things segregate out by Mendelian rules, and they're straightforward to study. But adult genetics and the genetics of all these things that kill us, like heart disease and cancer and congestive failure, turn out to be very complicated. And as a result, we identified the fact that there are many, many components, but made relatively little progress in understanding the complexity of these adult machines. Oligogenics, of course, is the thing that bridges the two, since we are, after all, one genetic system. Now, I'm not going to talk about this side of the graph because really because of what I said before, but I'll acknowledge that there is an enormous amount of data that is being used in quantitative studies because of the large numbers of GWASs and large sequencing cohorts that have been established throughout international studies. So many of you have heard of the UK Biobank, Genomics England, the TopMed program, the NIH's Complex Disease program, and Lydia mentioned the All of Us program, where we're reaching towards getting data from 1 million participants plus their medical records, all in this big cauldron, to try and solve some of their health issues. And that program is ongoing. There's about 300,000 genomes completed now, records assembled, and that will speed discovery. But despite all this, and despite everything else anyone will tell you about complex disease studies and about studies of these adult conditions, there is relatively little clinical utility from the genetics of their study at this time. And that's our conundrum. So let's turn back to this more simple problem and ask, can we, through this gateway, understand more about individual genes and maybe bring up an understanding that will compound and inform us more on common disease. Now, how many genes are there? There's about 20,000. How much do we know about them? Well, we know an association between a gene mutation and a phenotype for a little under 5,000 of those about now. That is about, these numbers are straight from the public database in the last week. That's a little less than half of the cases that we think for other reasons could be solved if we were able to do more systematic gene studies to make these associations. We don't know so much about the other half of all genes because some of them may not be solvable, they may be genetically lethal, or they may have no impact or very little impact in overall biology. But we can ask ourselves the question now, can we, through using better methods than we have in the past, solve the rest of these genes and use those to inform common disease? Well, here's how we do things today for the most part. And I'm calling this mode 1 because I want to show you sort of mode 1, mode 2, mode 3 as we move through levels of complexity and get more quantitative. Mode 1 is essentially sort of house by house. That is, we take individual families and we use the state-of-the-art sequencing methods to sequence pedigrees and affected individuals to determine if there's a genetic anomaly that segregates appropriately with the disease. In this case, a de novo mutation occurring in this individual that wasn't in the parents that indicates that gene might be the gene that underlies that syndrome. This is actually my grandniece, and this is one of the first ones that we did when we developed these new methods. So in that case, we found fairly quickly 3 other children with the same disorder, or the same general phenotype, the same gene mutated, and so we're able to establish that as a new genetic syndrome. So using that as a seed now, we have several hundred families now that have the same disorder. We have laboratory studies going on. We've built on the understanding around that single gene. And this is the story that's been repeated hundreds and hundreds of times in Mendelian disease biology. So this is a little out of date, but 2018, but this same curve has gone on for about every 30 people that get sequenced in these Mendelian gene discovery programs. We find a new gene, a new association between a gene and a phenotype. And that sort of adds to our catalogue of knowledge of biology based on single gene genetics. That actually sounds pretty good when you think about it, you know, just 30 sequences to get one person, but the whole enterprise is actually much slower than we would like. It did get us from 3,000 to 4,700 or so of these associations over 8 years across the whole globe, everybody's programs. But remember, we've still got about that amount to go. another 4,700 to go. That's just too slow. So we need to accelerate this mode, this one-by-one mode. So in thinking about that locally, we realised that we had accumulated in our gene studies a lot of what we call unsolved cases. That is, genetic data from individuals who potentially could have been studied to completion to understand the genetic disease they had, the association between the gene and the disease, but we simply haven't got there yet. Perhaps we hadn't looked deeply enough in that family, or we didn't have enough families with the overlapping phenotype, or the phenotypes were just too heterogeneous. But when we added up all the resources we had from these different things around the chart, our research rich component, the clinical samples coming in, our affiliates around the medical centre and the hospital system, and the dedicated programs, it's a lot of samples. So there's some more there from the diagnostic lab. So Adam Hansen, a student who graduated about a year ago, he undertook to look naively at these samples and ask himself, could we just take the genotypes themselves? Forget about where the samples came from, forget about the family history, forget about the clinical picture. Could we just look at the genomes and extract from those knowledge about whether or not there were disease genes that were indicated? So essentially, he took 18,000 unsolved cases, the genomes, and he put them in what was then a state-of-the-art environment. This is like a Hadoop structure with file structures that were not requiring very detailed structure matching between the files, Parquet files, and then simply searching those and doing parameter sweeps to see if he could characterise disease genes in individuals using known disease genes as a calibration for his methods. So he would go through those and establish gene lists and do this comparison and ask, was he finding new disease genes in this process? And the short answer there is that that worked pretty well. Is the trajectory for the disease genes for some of these parameters that were measured, and I won't go into details of what that is. And here's an unknown set of genes. And indeed, he was able to eke out a collection of around 150 genes which had not previously been associated with disease, just from looking at the genotype patterns in this fairly heterogeneous collection that we had. So it's pretty successful. Many of these were validated as the study continued, and since then they've continued to be validated. So it's not a bad way to mine out more disease genes. But when you think about it, it's still not scalable. It's still not enough to really push us into this state of knowing what all genes do. The conclusion from this is we actually needed to add more data at different levels to expand that study and to start to think more about the whole individual, including their phenotypes. But stepwise, the next thing I want to tell you, I'm calling it mode 2 here, mode 3, is the idea of looking at the variant collection in the genes themselves. And I'll just mention this kind of parenthetically. So this is the idea that if we simply study every gene, the ones we don't know, in a model system, and we make libraries that mutate every single base, every single nucleotide in each gene, and then we challenge that library with some kind of phenotypic selection, maybe we can distinguish the benign variants from the variants that damage the gene. Now, with CRISPR and other genetic editing techniques, it's become possible to construct those libraries. And then several years ago, Greg Findlay demonstrated this process in a breast cancer gene model, where the assay was to take a cell line that is haploid, and you ablate the gene you're interested in that case the breast cancer gene, and then you substitute it with all these different variations of the breast cancer gene, and then you ask that collection of cells who will grow. And the cells that grow have the gene that works, the cells that don't have deleterious mutations. So this system's now being extended by Leah Starita, and Jim Lupsky and myself, student Moes, has gone to Seattle for a year now to help to advance that system and to work on it. So we've got an assay with complete libraries, growth in this cell line, and you simply take the cells at different times and sequence them and see which variants have grown out. And the ones that are benign grow well and are seen in this, they're seen there at later days in the cell culture. The ones that are pathogenic on the left there do not prevail. So Moes has been studying PALB2, which is related functionally to the breast cancer gene. And indeed, the assay works. He's got benign variants mapped from different regions of the protein, and then he's got pathogenic variants from other regions. He's got some exceptions to that that are being deep dived into to understand if they have some intricate biology. And in general, he's showing that this assay can exhaustively assay those genes. The pathogenic variants do correlate very well with the impact in the clinic of those genes, and the method works great. But the trouble is it's very labour intensive. Okay, it's taking a student to do a couple of genes. It's being scaled. This will bring a lot more characterisation to these different individual genes, but it's not going to get us there in a hurry. So we still need to do something else. And of course, the something else is to delve into clinical data, phenotype data, and to bring that aspect to these genetic analyses. Now, so as I just said, we've got the low-hanging fruit you saw in those last two methods, but we need to explore these phenotypic approaches. So how do we do it if we can't simply take medical records and throw them in a cauldron? Well, one answer to that is from work that began about a decade ago in our diagnostic lab, when it was realised that about 5% of the individual childhood disease cases that were being studied had the unusual phenomenon of more than one genetic disease in the child. Let me explain that clearly. This is a distinct phenomenon. It's not like, I'm not talking about complex disease here. You imagine a child with cystic fibrosis and a child with muscular dystrophy, distinct, well-characterised mutations, well-characterised genes, well-characterised diseases. Statistically, it's possible a child would have both. So these are cases where children actually had mutations in well-characterised disease genes, clearly driving two individual, what could be individual Mendelian diseases. And the stunning thing for us in those early days was the frequency of this. Amongst about a third of the cases that could be diagnosed, there were 5% which had this phenomenon. There are even children with 3 different genetic diseases going on at once, and one case with 4 individual Mendelian diseases going on. So remember the frequency here. If you go and sequence everybody, you might find about 1% of us have some kind of genetic signal that would be in the genetic disease class, right? And in children, when these are expressed, so that's about the number, about 0.9% of children have these kind of disorders. If you imagine a child who has more than one of these disorders, you can imagine that they would fall into 2 possible categories. Those where the individual diseases have quite different phenotypes. Here's a case of oculocutaneous albinism that affects the eye and skin pigmentation. A second disorder in the same child has a connective tissue disorder, right, affecting joints and some of the arteries. But you can imagine other instances where the phenotypes generally overlap. Here's 2 genes which separately cause different kinds of epilepsy and different kinds of developmental delay. So these represent 2 quite different categories, distinct and overlapping modes of disease. Jennifer and her team asked themselves about this. Could they use phenotypic classifiers to distinguish these 2 types of multiple disease? Really as a handle to understand how they could better quantitate these phenotypes, explore them in rare disease, and use them to build more elaborate disease models. So I think many of you know about the human phenotype ontology, HPO. The idea is you have a series of classifiers with ascending weight. Some of them are more general, and as you go down the tree, you get more specific. If you want to build a score, then of course the score increases up the tree and the weight is lower from elements down the tree. And what she and the team did was to take these cases of either overlapping or distinct disorders and then have human experts classify them and then separately build a set of ontology-based scores and use those to compare the individual cases and then to rank them in this comparison. And sure enough, they found they could weight differently those cases that were overlapping, those cases that were distinct. So this is, if you're in this area, it's actually a big breakthrough. Suddenly you've got a handle on some of these phenotypes that can tease apart some of these otherwise complicated childhood disorders. Let me show you an example of how that can work. So this is a generalised disorder called Robinell syndrome that has spectrum of phenotypes and has about a half a dozen genes or so that have been assigned to that particular syndrome. And these are the genes here and the percentage of individuals in the local cohort that contribute to that disorder. When the phenotypes from this disorder were classified in this way and then built into a pairwise comparison, they gave this expected similarity down the diagonal here. But what was particularly encouraging is if you look in detail at these clusters, there are regions that correspond to the individual genic cause. And that goes on through the different genes that underlie the syndrome. Again, the encouraging thing here is a handle on the phenotypes and a scoring system that can begin to unpack these data. And I'll give you a final case, which is quite dramatic, I think, of a single individual using more or less the same approach. So here's a child who was diagnosed with 4 diseases, and you can see the different areas. Here's a sort of physiology map of the different disorders, some overlapping, some distinct. When the team assessed the phenotypes of the child and compared that to the expected phenotypes from the individual genes, here's the child, this is the reference number for the child. These are the different genes underlying the disorder, and you see presence or absence of the phenotype. When you aggregate all of these elements up from the individual disorders and you put them together, you get the complete phenotype of the proband. So all of this goes towards the idea that if you use these phenotype weights, you can begin to dissect these simple diseases and really get a handle on these relationships. This is useful for more gene discovery from the childhood disease cases, but also offers opportunity ultimately to leverage us back into common disease to use phenotypes to help understand the mysteries of the complexity of adult disease. Well, to do that, of course, we need more phenotypic information, and as I think underlies much of what everybody wants to do, we ask ourselves, why can't we extract that information from the electronic health record? And so the next question we asked ourselves that is, where does one begin? Right? You can't boil the ocean. How do you begin to get into that? Well, to do that, our entree is through a program we call HeartCare. Now, I've just spent the last 20 minutes telling you about the difference between complex disease and simple disease. HeartCare is a program where we're looking at adults to ask, can we find genetic risk markers for them that affect their cardiovascular care? Now, here's our dilemma, and it's the dilemma I mentioned right at the beginning of my comments. That is, we don't really have enough information about genetics to offer a genetic test for adults that considers all the complexity of adult genetics. But we do know that a significant fraction of adult risk is also conveyed by single gene risk factors, simply that haven't been seen yet through their life, but still engage them as adults. Now, as I mentioned before, if we go out and just sequence people randomly, we might find that at a rate of about 1%. And I can tell you that discovery at the rate of 1% is not enough to sustain a clinical genetic screening program. You cannot get anybody to be enthusiastic about sequencing 100 people just to find one individual who might have a genetic risk factor. So we built this adult program here, and we biased it by not getting random individuals. Instead, we went to clinics where we might have enriched for some kind of more severe genetic condition. And in this case, this was the ambulatory Baylor cardiovascular care clinics. Now, we did not go into the hospital to find people with very severe disease, so there's a very wide net that we posted, but it was people who had some kind of a prior risk for cardiovascular disease. And we offered them the chance to participate. They would get back genetic results in this nice brochure, and it wouldn't cost them anything. Here are the genes and the conditions that we studied. We actually, in the beginning, ran a gene panel. Later, we're doing whole genome sequencing and taking virtual panels, but this is a gene panel. And then we started to enroll. We actually stopped during COVID, a little over 600, primarily because of COVID. And then did our complete analysis, returned all the data to everybody. Here's the steps that were involved. If you came to one of the Baylor clinics, the physician or physician's assistant would talk with you, you'd get consented, give a specimen, and then at a subsequent visit, you'd get a result. You'd get it in the mail if it was a negative result, but if it was positive, you'd get that'd go through your record, and your physician would give it back to you, and there was reflexive genetic counselling afterwards. So our motivation, of course, was to get the data for research. This is the attraction for the patient. Here's a genetic report that everybody got. It told them if they had a severe genetic condition, that is something that was something like a Mendelian condition, a single gene condition that might affect them dramatically. And the rest of the stuff was more gravy. There's a thing called the polygenic risk score, which I won't talk about today, but which is a generalised genetic risk factor estimate, telling you if you're in the top 10% or not. One particular lipid marker and some drug reaction markers. Now, this was the result that we got from that. And the most astounding thing in this whole chart here is down here in this column. The 64 of the 640 individuals had a likely pathogenic or pathogenic mutation in a single gene that conveyed high-risk cardiovascular disease. Now, that's the number, nearly 10%. You can compare to that 1% in the, if we sequence the general population, we just 1%. This was a high enrichment for that kind of individual. These other folks here, this is the aggregate of all genetic signals. Here's those other things, and pharmacogenomic risk very high. We say from this study around about 30% of people received information the physicians found actionable. So the overall result of this study was that there was more genetic action and more stuff to be told to people who came through these clinics that enriched for this kind of population, and many of those had single gene defects. So enrichment works. In this case, the enrichment is having the kind of clinic that attracts the person. So that's fine. But it's not the kind of enrichment that you can apply on a population-wide scale. So the dream following this experiment was, could we find other factors that would actually enrich for the population that most benefited from genetic testing before we did the genetic testing? Is there somewhere in the medical record a series of terms that, if matched properly, that they would tell us that a person had an enhanced risk for a genetic condition, therefore it was worthwhile to do the genetic test on them? If only there was a miracle computer technology that could scour medical records and find such signals. That's our thinking, okay? So the thinking then is not to do the magic cauldron experiment and throw everything in and hope to come out with something mature, but take the humility, take the lesson, do something focused, and put in minimal key data, the genetic test result for the training and the record, to see if we could figure out what would be the characteristics of these people, and would indeed we be able to take the Baylor records of hundreds of thousands of people and tell 10,000 of them or 20,000 of them come and get a genetic test, because that we could afford. Now, we've embarked on that with specific fervour since Lydia called me and said, would I come to the conference? I've accelerated that part of the program. And have been working through these familiar problems. How do you get the data access? How do you deal with the data structure issues, even before you begin mining that and asking what have you found from your individual models? And I suspect many of you in the audience now are getting familiar with what I'm showing, because you're starting to see the sort of stuff you get out of medical records. And so we've been able to get the data from our heart care population, which is kind of small. And as you know, in addition to those semi-structured data that I just showed in the last slide, most of the juicy stuff is in the clinical notes, which are an absolute mess. Okay? So, like many, we've tried to use natural language processing to extract from this morass data that could then be used to feed into methods to build models. And we've seen many of the problems that I'm sure many of you have also seen, such as why is this coming up twice from the same extraction? Anyway, here's the meat of what I can tell you, which is not very inspiring. Well, let me say it flat out. Our training set of 500 or so people with 40 of our true positives and our test set, we're seeing low predictability. We're seeing, I guess this is good specificity, right? The true negatives are there, but we're only seeing a little bit of the predicted results. But it's early days. We see that with sort of these generic methods you can find online. Javid has run Decision Tree. It was late last night, he sent me a tree and he said, I just don't understand why familial hypercholesterolemia is at 2 nodes in the tree until you look at the spelling. And that starts to explain why. So the message here, of course, is that this is the right aspirational goal. I think this is the right entry point for this kind of a study. We're early on in that and we have some way to go, but I think the value of that is extremely high. So, what can I conclude from my comments? We do, I think we'd say we are using AI methods in niche applications for prediction quite successfully. The big challenge we have is to complete this disease gene catalogue, build these complete models where we understand something about the phenotype for every single gene. We know now that we can't just keep doing that gene by gene. We need to start to explore clinical records and do this through full clinical integration. But we are certainly not there yet, despite our optimism. Now, of course, all of this work's done by a whole lot of people, and I showed many of them on the way through. I want to acknowledge Donna, who gave a talk in your workshop yesterday, who's been running the Genome Centre for 25 years, my co-director, Eric Bowinkle from UT, and Eric Benner's here in the audience, and I think Ji Hong's here in the audience, and I'm sorry if I forgot anyone else from our team. But thank you.
How many people do you think we have to sequence to create an accurate disease model?
I think if we had complete, the ideal phenotype from basically everyone who's been done so far, we could go to the 90% level.
Okay.
I think the gaps in our information and our integration and our completion for these individuals are so great as to hamper. We're just picking things out of the morass from very sparse data right now.
Some questions?
EHR remains one of the most confusing place where there are thousands of people writing in a single person's chart. Often there's a really copy-paste kind of component too. In your experience, how much it is hindering? A same patient goes to different healthcare systems. Sometimes they're connected, sometimes not. But whatever you have so far researched, what's your experience so far with this messy EHR data? Is this enough to make meaningful results?
No, there's a fundamental issue, of course, that the clinical ascertainment is usually different from the discovery ascertainment. That's probably the big one. I mean, if you go in, with a broken leg, they're not going to look at your kidney function, and yet, you might have some systems problem that the individual could contribute to discovery in. So, is there a vehicle for comprehensive phenotyping that is in any way imaginable as practical to be embedded in a medical record? That's kind of a first question, and I think the answer may unfortunately be probably not. So that's a big obstacle. But then I think at the other end of it, the simple heterogeneity of the clinical notes, big obstacle, and the reluctance of individual caregivers to rely on pull-down menus when they want to inject their own subtle interpretation on top of the pull-down menu. You sound like the kind of guy who could solve this. Is that?
I wish. I'm a clinician and I deal with those records, and we do.
Do you agree with my comments?
I agree with you. I think we actually just published a research in central sleep apnea, and that was one of the big concerns, is the diagnosis which we are picking up from those charts, how reliable they are. And some studies were saying it's about 60% reliability. So it's a big problem.
You may have to push the button to get heard.
This one?
I'll go green.
Can you hear me?
Yeah.
Very informative presentation for clinicians like me. One thing that we encounter when we clinically see the patient is the genes you may be able to know, but the effect of environment is not documented. So we would see patients that have smoked for many years, but we don't know how much they smoke. We just estimate. The second question is that the research that he was mentioning, we have come across insults that traditionally we consider them as harmful, like hypoxia. And then at the same time, we will see a large number of the patients when we look at the databases that hypoxia, because it was not very severe, has actually caused a survival advantage rather than increasing the mortality. So that interaction is very important, which brings to the point that you say, if we can identify the phenotype and say this is the phenotype that differs from that maybe then we can basically more toward identifying the genetic background on that one.
Yeah, I think your comments remind us of the complexity and of the contribution of the environment, especially in these adult conditions that are multigenic, many small subtle contributions that are more variable with the environment. And with that as a background, how can you reduce that to a single element of clinical practice? I think that's a reasonable objection. The pure geneticist answer to this is if we gene by gene begin to understand expressivity and penetrance, the way, when does the gene really do the damage and how much damage does it do, then we start to look at the environmentally measurable components that correlate with those. Maybe we can make some progress in predicting environmental impact on the genetics. Okay, thank you.
Thank you so much.